Next up, we have oral presentation 7, Moreno Valley, California, presenting a Mission 13 flight experiment.
Our experiment is the growth and development of sustainable brine shrimp in microgravity. We are from Moreno Valley, California, Palm Middle School, Moreno Valley Unified School District. Our co-principal investigators are Vince Moreno,
Vincent Reese, and Aiden Alvarez.
Our co-collaborators are Andre Portlock and Doran Aries, which weren't able to make it today. And our teacher facilitator is Mrs. DiCantonio from Palm Middle School.
Our purpose statement. We will examine how brine shrimp hatch and develop in response to microgravity. We believe that our experiment will contribute to their use as a sustainable food resource. The brine shrimp can adapt to harsh conditions and could be used as the primary consumers of the food chain in raising fish and other crustaceans in an aquaculture setup in space or on another planet. We hope that our experiment is successful and can be used for further research in this area.
The brine shrimp life cycle. Hatched brine shrimp go at least go through 14 stages of development, and at each stage they shed the old exoskeleton and grow a larger one, also known as molt. The whole process takes from 8 days to 6 weeks depending on environmental conditions. We evaluate our microgravity exposed and ground truth brine shrimp for any differences in the stages of development, the number of brine shrimp at each stage. Why we chose brine shrimp? We chose brine shrimp because we found out that they are a good food source because they contain 37 to 71% protein. They can also be fed inexpensively with food sources such as yeast, wheat flour, egg yolk, and soybean powder. They produce dormant eggs, also known as cysts, that can be stored for long time periods and hatch when rehydrated with salt water. They are adapted to live in extremely stressful, salty lake habitats. They normally hatch within 2 days of rehydration and can develop to adults as little as 8 days.
Our mini lab setup. In our volume 1, there will be 3 milliliters of purified salt water adjusted to pH 8 at ambient temperatures. In our volume 2, there will be approximately 200 brine shrimp eggs at a tiny amount of less than 1 milligram of spirulina blue-green algae. The total volume is around 3 milliliters. In our volume 3, 2.4 milliliters of 10% buffered formalin at ambient temperatures to eliminate the brine shrimp. Our timeline of crew interaction. 2 days after arrival, the astronauts will unclamp clamp A and shake gently for 5 seconds to mix the salt water and with the brine shrimp eggs. 2 days before undocking, they will unclamp clamp B and shake the formalin with the brine shrimp eggs for 10 seconds.
Our post-flight analysis. After our International Space Station experiment has returned to our classroom, we will compare the brine shrimp exposed to microgravity to our ground truth experiment. The brine shrimp will be loaded into an identical FME mini lab that remains in the classroom and is manipulated according to the same procedure as the experiment that flew on the ISS. Specifically, we will analyze both preserved samples for the percentage of brine shrimp hatched, the differences in development, and the differences in size at similar stages of development.
This is our bibliography. And our acknowledgements are Principal Dr. Harris and our district facilitator, Amy Glancy, and our partners are Moreno Valley Unified School District, California, Spainsboro. And that is all for our presentation.